Today we will discuss the continuation of the competency on molecular biology, RNA synthesis, processing and modification. So now coming to RNA molecules. These RNA molecules are processed before they become functional. In prokaryotic organisms, the primary transcripts of messenger RNA encoding genes begin to serve as translation template even before their transcription has been completed. This can occur before the site of the transcription is not compartmentalized into the nucleus as it is in eukaryotic organisms. Thus, transcription and translation are coupled in prokaryotic cells. Consequently, prokaryotic messenger RNAs are subjected to little processing prior to carrying out their intended function in protein synthesis. Indeed, appropriate regulation of some genes relies upon this coupling of transcription and translation. So, these prokaryotic ribosomal RNA and transfer RNA molecules are transcribed in units considerably longer than the ultimate molecule. In fact, many of the transfer RNA transcription units encode more than one transfer RNA molecule. Thus, in prokaryote, the processing of these ribosomal RNA and transfer RNA precursor molecule is required for the generation of the mature functional molecules. Nearly all eukaryotic RNA primary transcripts undergo extensive processing between the time they are synthesized and the time at which they serve their ultimate function, whether it be as messenger RNA or microRNA or as a component of the translation machinery such as ribosomal RNA or transfer RNA. Processing occurs primarily within the nucleus. The process of transcription, RNA processing, and even RNA transport from the nucleus are highly coordinated. Indeed, a transcriptional coactivator term SAGA in yeast and F/CAP in human cells is thought to link transcription activation to RNA processing by recruiting a second complex term TREX to transcription elongation, splicing, and nuclear export. This transcription export represents a likely molecular link between transcription elongation complex, the RNA splicing machinery, and nuclear export. This coupling presumably dramatically increases both in fidelity and the rate of processing and movement of messenger RNA to the cytoplasm for translation. So coming to the coding portions, exons of most eukaryotic genes are interrupted by interons. So the RNA sequence that appear in mature RNAs are termed as exons. In messenger RNA encoding genes, exons are often interrupted by long sequences of DNA that neither appear in mature messenger RNA nor contribute to the genetic information ultimately translated into the amino acid sequence of a protein molecule. In fact, this sequence often interrupt the coding region of the structural genes. This in human messenger RNA encoding genes, exons average -150, while interons are much more heterogeneous, ranging from 10 to 100 to 30,000 nucleotides in length. The interon RNA sequence are clipped out of the transcript, and the exons of the transcript are approximately spliced together in the nucleus before the resulting messenger RNA molecule appear in the cytoplasm for translation. So interons are removed and exons are spliced together. So several different splicing reaction mechanisms for interon removal have been described. The one most frequently used in eukaryotic cells is here. Although the sequence of the nucleotide in the interons of the various eukaryotic transcripts and even those within a single transcript are quite heterogeneous, there are reasonably conjugate sequences at each of the two exon-interon junctions and at the branch site, which is located 20 to 40 nucleotide upstream from the 3' splice site. A special multi-component complex, the spliceosome, is involved in converting the primary transcript into messenger RNA. Spliceosomes consist of the primary transcript, 5 small nuclear RNAs, U1, U2, U4, U5, and U6, and more than 60 proteins, many of which contain conjugate RRM, RNA recognition, SR, and serine arginine protein motifs. Collectively, the 5 small nuclear RNAs and RRM or SR containing protein form a small nuclear ribonucleoprotein termed as small nuclear RNP complex. It is likely that this penta SNRNP spliceosome forms prior to interaction with messenger RNA precursors, and small nuclear RNPs are thought to position the exon and intron RNA segment for the necessary splicing reaction. The splicing reaction starts with a cut at the junction of the 5' exon donor or left and intron. This is accomplished by a nucleophilic attack by an adenyl residue in the branch point sequence located just upstream from the 3' end of this intron. The free 5' terminal then forms a loop or layered structure that is linked by an unusual 5' to 2' phosphodiester bond to the reactive A. This adenyl residue is typically located 20 to 30 nucleotide upstream from the 3' end of the intron being removed. The branch site identifies the 3' splice site. A second cut is made at the junction of the intron with the 3' exon donor or on right. In this second trans-esterification reaction, the 3' hydroxyl of the upstream exon attack the 5' phosphate at the downstream exon intron boundary, and the layered structure containing the intron is released and hydrolyzed. So the 5' and 3' exons are ligated to form a continuous sequence. So alternative splicing provides for different messenger RNA. The processing of messenger RNA molecules is a site for regulation of gene expression. Alternative patterns of messenger RNA splicing results from tissue-specific adaptive and developmental control mechanism. Interestingly, recent studies suggest that alternative splicing is controlled, at least in part, through chromatin epigenetic mark. This form of coupling of transcription and messenger RNA processing may either be kinetic or mediated through interactions between specific histones, PTMs, and alternative splicing factor that can load onto nascent messenger RNA gene transcript during the process of transcription. Faulty splicing can cause disease. At least one form of P. thalassemia, a disease in which the P globin gene of hemoglobin is severely underexpressed, appears to result from a nucleotide change at an exon-intron junction, precluding removal of the intron and therefore leading to diminished or absent synthesis of the F3 chain protein. This is a consequence of the fact that the normal translation reading frame of the messenger RNA is disrupted by a defect in the fundamental process of RNA splicing, undergoing the accuracy that the process of RNA splicing must be maintained. So now coming to alternate promoter utilization provides a form of regulation. So tissue-specific regulation of gene expression can be provided by alternative splicing, by control element in the promoter, or by the use of alternative promoters. The glucokinase gene consists of 10 exons interruption by 9 introns. The sequence of exon 2 to 10 is identical in liver, pancreas, or pancreatic 13 cells. The primary tissue in which this glucokinase protein is expressed. Expression of this glucokinase gene is regulated very differently by two different promoters in these two tissues. Both ribosomal RNA and most transfer RNA are processed from larger precursors. So RNA can be extensively modified. Essentially, all RNAs are covalently modified after transcription. It is clear that at least some of these modifications are regulated. So messenger RNA is modified at 5' and 3' ends, and poly A tails are added to the 3' end of messenger RNA molecules in a post-transcriptional processing step. The messenger RNA is first clipped about 20 nucleotide downstream from an AAU-AA recognition sequence. Another enzyme, poly A polymerase, adds a poly A tail which is subsequently extended to as many as 200 A residues. The poly A tail both protects the 3' end of messenger RNA from 3' to 5' exonuclease attack and facilitates translation. The presence or absence of this poly A tail does not determine whether a precursor molecule in a nucleus appears in the cytoplasm. because all poly-A tail nuclear membrane RNA molecules do not contribute to cytoplasmic messenger RNA, nor do all the cytoplasmic messenger RNA molecules contain poly-A tails. Histone messenger RNA are most notable in this regard. MicroRNAs are derived from large primary transcript through specific nucleolytic processing. So RNA editing changes messenger RNA after transcription. The central dogma states that for a given gene and gene product, there is a linear relationship between the coding sequence in DNA and the messenger RNA sequence and the protein sequence. Transfer RNA is extensively processed and modified. So this RNA can act as a catalyst. In addition to the catalytic action served by the small nuclear RNAs in the formation of messenger RNA, several other enzymatic functions have been attributed to RNA. The ribosomes are RNA molecules with catalytic activity. These generally involve trans-esterification reactions. Most are concerned with RNA metabolism, splicing, and endoribonuclease. Recently, ribosomal RNA component has been implicated in hydrolyzing an amino acyl ester and thus to play a central role in peptide bond function, peptidyl transferases. These observations made using RNA molecules derived from the organelles from plant, yeast, viruses, and higher eukaryotic cells so that RNA can act as an enzyme and have revolutionized thinking about enzyme action and the origin of life itself.